Have you ever looked up at the night sky and wondered if we are alone in the universe? This is one of the biggest questions humans have ever asked. For thousands of years, we have told stories about other worlds and the creatures that might live there. One of our closest neighbors, the planet Mars, has always captured our imagination. It's a reddish dot in the sky that looks both familiar and mysterious. Scientists and dreamers alike have long wondered if life could exist on its dusty surface. 50 years ago, in 1975, NASA launched a mission that would try to answer this very question. It was called Viking, and it changed everything we thought we knew about Mars. The Viking missions were not just another space probe. They were a monumental effort, a bold and ambitious plan to do something no one had ever done before: land a fully functional laboratory on the surface of another planet. This was the first time humanity would directly search for extraterrestrial life. Before Viking, our views of Mars came from telescopes on Earth or from spacecraft that flew by without stopping. These gave us tantalizing glimpses, but they couldn't tell us what was happening on the ground. The Viking missions were designed to get up close and personal, to touch, test, and analyze the red planet in a way that was previously only science fiction. Imagine the excitement and the tension back then. Sending a spacecraft millions of miles through the harsh environment of space is incredibly difficult. Landing it gently on an alien world is even harder. The Viking landers were pioneers, robotic explorers, venturing into the unknown. Their journey represented a huge leap in technology and our quest for knowledge. They were our eyes, our hands, and our scientific instruments on a distant world. The data they sent back would either confirm our greatest hopes or present us with a puzzle that would take decades to begin to solve. The story of Viking is a story of human curiosity at its finest. On July 20th, 1976, the world held its breath as Viking 1 sent back its first picture from the surface of Mars. It was a historic moment, happening on the 7th anniversary of the first human moon landing. The image that appeared was breathtaking. It showed a rocky, reddish landscape under a pale pink sky. For the first time, we weren't just imagining Mars, we were seeing it. This wasn't a drawing or a guess, it was a real photograph from another planet. The image was published on the front page of newspapers around the world, making Mars a real place in the minds of millions. This was our first true foothold on the red planet. The Viking program was not just one spacecraft but two identical sets of explorers. Each set was made of two parts, an orbiter and a lander. Think of the orbiter as a scout flying high above, and the lander as the scientist on the ground. NASA launched Viking 1 in August 1975, followed by its twin, Viking 2, just a few weeks later. These spacecraft embarked on a long journey, traveling for nearly a year to cross the vast distance between Earth and Mars. Their mission was one of the most complex and ambitious ever attempted in the early days of space exploration, pushing the boundaries of what was technologically possible. After arriving at Mars in the summer of 1976, the orbiters got to work. Their first job was to map the planet's surface in incredible detail. They were looking for the perfect safe place for their landers to touch down. This was a critical task. Mars is covered in craters, canyons, and giant volcanoes, so finding a smooth, flat area was essential for a successful landing. The orbiters acted as communication relays, sending commands from Earth to the landers and transmitting precious data back home. They also studied the Martian atmosphere and weather, giving us our first comprehensive look at the planet's climate from above. The landings were the most nerve-wracking part of the mission. Viking 1 separated from its orbiter and began its descent on July 20th. 1976. It entered the thin Martian atmosphere protected by a heat shield, deployed a parachute to slow down, and then fired its retro rockets for a gentle, controlled touchdown in a region called Planitia, or the Golden Plain. A little over a month later, on September 3, 1976, Viking 2 successfully repeated this incredible feat, landing farther north in a place called Planitia. These were the very first successful soft landings of fully operational spacecraft on Mars, a monumental achievement for NASA and the entire world. Once safely on the ground, the Viking landers became our robotic representatives on Mars. They were built to last. The Viking 1 lander operated for over 6 years, sending back data for 2,245 Martian days, or sols. This set a record for the longest operating spacecraft on the Martian surface, a record that stood for decades until it was finally broken by the Opportunity rover in 2010. The landers were sturdy and reliable, a testament to the brilliant engineering that went into their design. They were our first long-term residents on the Red Planet, diligently working day after day to unlock its secrets. The Viking missions had a list of ambitious goals, and taking pretty pictures was just the beginning. The primary and most exciting goal was to search for signs of life. This was the first, and to this day, the only mission to directly test for living microorganisms on another planet. Scientists at the time thought that simple life forms, like bacteria, might be able to survive in the harsh Martian environment. The landers were equipped with a sophisticated miniature biology laboratory designed to scoop up Martian soil and run experiments on it. Everyone was eager to find out if Mars was home to its own native life. Beyond the search for life, Viking was designed to be a comprehensive scientific station. The landers carried a suite of instruments to study geology, meteorology, and chemistry. Their cameras provided stunning, high-resolution panoramic images of the landscape, revealing a world of red rocks and fine-grained dust, stretching to the horizon. These images help geologists understand the processes that have shaped Mars over billions of years, like wind erosion and ancient floods. The landers also had a weather station that measured temperature, wind speed, and atmospheric pressure, giving us our first daily weather reports from another planet. These reports revealed a dynamic world with changing seasons. The Viking orbiters also had a crucial role in understanding Mars as a whole planet. While the landers explored their small patch of ground, the orbiters circled above, mapping nearly the entire surface. They discovered that Mars once had a much denser atmosphere than it does today, hinting that the planet might have been warmer and wetter in its distant past. They also solved a long-standing mystery: why the Martian sky is pink. It's because strong winds constantly lift fine red dust particles into the atmosphere, scattering sunlight and giving the sky its signature color. This dust also explains the seasonal changes in atmospheric pressure, as carbon dioxide freezes at the poles in winter and turns back into gas in summer. At the heart of the landers was a robotic arm that could dig trenches in the soil and deliver samples to the onboard experiments. One of these experiments was a gas chromatograph mass spectrometer, or GCMS. Its job was to analyze the chemical composition of the soil and search for organic molecules. Organic molecules are the carbon-based building blocks of life as we know it. Scientists expected to find them on Mars, even if there was no life, because meteorites containing organic compounds constantly rain down on the planet. Finding them would be a key piece of the puzzle, but what the GCMS found, or rather, didn't find, would become one of the biggest mysteries of the mission. The Viking landers carried 3 separate biology experiments, each designed to look for life in a different way. The first and perhaps most famous was the labeled release, or LR, experiment. The idea behind it was simple, but clever. If there were tiny living microbes in the Martian soil, they would probably need to eat to survive, just like living things on Earth. The LR experiment was designed to offer them a meal and see if they ate it. The meal was a liquid nutrient, like a broth or a soup, that was specially prepared. It was a mix of simple organic compounds that many Earth bacteria love to consume. What made this experiment so clever was that the nutrient soup was tagged with radioactive carbon-14. This radioactive tag acted like a tiny tracker. When microbes on Earth eat nutrients, they release waste gases as part of their metabolism. One of the most common waste gases is carbon dioxide. So the LR experiment was set up to detect if any radioactive carbon dioxide gas was released after the nutrient was added to the Martian soil. If the radioactive tag showed up in the gas above the soil, it would be a strong sign that something in the soil was metabolizing, or eating, the nutrient. When the results from the labeled release experiment came back from Mars, they were astonishing. As soon as the radioactive nutrient was added to the Martian soil sample, there was a sudden and significant spike in the amount of radioactive gas detected. The response was exactly what scientists would have expected to see if the soil were teeming with microorganisms. The initial data looked like a positive result for life. The principal investigator of the experiment, Dr. Gilbert Levin, was convinced that he had discovered living microbes on Mars. It was a thrilling and potentially world-changing moment. However, the excitement was tempered by a few puzzling details. When a second dose of the nutrient was added to the same soil sample a week later, there was no new release of gas. This was strange. If living microbes were present, you might expect them to continue eating. Also, when another soil sample was heated to a high temperature to sterilize it before adding the nutrient, there was no gas release at all. While this supported the idea of life, since heating would kill microbes, it also left room for another explanation. Could a non-biological chemical reaction be mimicking the signs of life? The LR experiment gave a tantalizing yes, but the other experiments would soon complicate the story. Alright, let's dive into Section 5. Experiment 2, which is famously known as the carbon assimilation conundrum. So the second biology experiment on the Viking landers was called the carbon assimilation experiment, also known as the pyrolytic release experiment. Now, this one, it took a completely different approach to looking for life. Instead of trying to detect metabolism by seeing what microbes breathe out, this experiment tried to see what they might breathe in. You see, on Earth, plants and some microorganisms perform photosynthesis. They take carbon dioxide from the atmosphere and use sunlight to convert it into organic matter to build their bodies. Pretty fascinating, right? The carbon assimilation experiment was designed to see if any Martian microbes were doing something similar. To test this, the experiment took a sample of Martian soil and placed it in a chamber filled with a simulated Martian atmosphere. Now, this atmosphere was special because it contained carbon dioxide and carbon monoxide made with radioactive carbon-14. The soil was then incubated under a lamp that mimicked the Martian sun for several days. The idea was that if any microorganisms were present and were fixing carbon from the atmosphere, they would incorporate the radioactive carbon into their cells. After the incubation period, the soil was heated to a very high temperature, to break down any organic matter and release the carbon. If the soil had captured the radioactive carbon, it would be detected when the sample was heated. But, here's the thing, the results of this experiment were far less dramatic than those of the labeled release experiment. There was a small amount of radioactive carbon detected in some of the test runs, which initially seemed interesting. However, the amount was very low, and the results were inconsistent. Most importantly, a similar amount of carbon was also fixed in a control sample that had been sterilized by heat before the experiment began. This suggested that the weak signal wasn't coming from a biological process. Scientists concluded that the small amount of carbon fixation was likely caused by a non-biological chemical reaction in the Martian soil itself. Perhaps some unusual minerals in the soil were reacting with the radioactive gases under the experimental conditions. Because the sterilized control sample produced a similar result, it was very difficult to argue that life was responsible for the signals seen in the carbon assimilation experiment. This result stood in stark contrast to the exciting, positive-looking data from the labeled release experiment. The search for life on Mars was quickly becoming a very confusing puzzle. The third and final biology experiment was called the gas exchange experiment. This one was designed to be a broad search for any signs of metabolism. It worked by monitoring changes in the gases above a soil sample. The experiment had two main stages. In the first stage, a small amount of Martian soil was placed in a sealed chamber, and a nutrient-rich liquid, playfully nicknamed chicken soup by the science team, was slowly added until the soil was wet. The instrument then carefully measured the composition of the gases in the chamber over a long period, looking for any changes that might indicate biological activity, like the release of oxygen, carbon dioxide, or hydrogen. When the nutrient broth was added to the Martian soil, something very unexpected happened. A large amount of oxygen was immediately released from the soil. This was a huge surprise. On Earth, some microbes produce oxygen, but this was a very rapid and intense burst of gas. Furthermore, the oxygen was released as soon as the soil got wet, even before the nutrients could have been metabolized by any potential life. This quick, chemical-like reaction made the scientists suspicious. It didn't look like a slow, biological process. It looked more like a simple chemical reaction. The experiment continued for months, but after the initial burst of oxygen, not much else happened. The gases in the chamber remained relatively stable, with no further signs of the metabolic activity that scientists had hoped to see. To investigate further, the experiment was also run in a humid mode, where the soil was exposed only to water vapor instead of the liquid nutrient. Even in this case, a significant amount of oxygen was released. This strongly suggested that the reaction was not with the nutrients in the chicken soup, but with the water itself. something in the Martian soil was reacting powerfully with water. The science team concluded that the oxygen release was not a sign of life. Instead, they proposed that the Martian soil must contain highly reactive chemical compounds, such as peroxides or superoxides. These are chemicals that can be formed by the intense ultraviolet radiation from the sun that bombards the Martian surface, since Mars has a thin atmosphere and no protective ozone layer. When these reactive chemicals come into contact with water, they can break down and release oxygen. This purely chemical explanation fit the observations perfectly, and provided a non-biological answer for the gas exchange experiment's strange results. The case for life on Mars was looking weaker. While the three biology experiments were giving confusing and contradictory results, another instrument on the Viking landers was conducting a different kind of search. This was the Gas Chromatograph Mass Spectrometer, or GCMS. Its job was not to look for active life, but to search for the chemical building blocks of life-organic molecules. Organic molecules are compounds that contain carbon, and they are essential for all life as we know it. Scientists were not just hoping to find them, they were fully expecting to. Even if Mars was lifeless, it should still have organic molecules. The reason for this expectation was simple. Space is full of organic compounds. For billions of years, Mars has been bombarded by meteorites and cosmic dust, many of which are known to be rich in organic molecules. These materials fall to the surface and should have accumulated in the soil over eons. So, when the Viking GCMS scooped up soil samples, heated them to release any volatile compounds, and analyzed the results, everyone anticipated finding at least some trace of these cosmic leftovers. This finding would have been a crucial piece of the puzzle, providing a starting point for understanding Martian chemistry. The results from the GCMS were a complete shock. The instrument found nothing, no organic molecules, at all. The soil samples from two different locations on Mars, thousands of miles apart, appeared to be completely sterile of any organic compounds, at least down to the parts per billion level that the instrument could detect. This was a profound mystery. How could a planet that is constantly receiving organic material from space have soil that is completely devoid of it? It just didn't make sense. Something on the Martian surface must be actively destroying organic molecules. This surprising lack of organics became the final nail in the coffin for the idea of life on Mars, at least for the Viking science team. The logic was straightforward. You can't have life without the building blocks of life. Even though the labeled release experiment gave a positive-looking result, it was now seen as an anomaly. How could there be living microbes if there were no organic molecules to build their bodies from, and no organic traces left behind from dead microbes? The official conclusion of the Viking mission was that there was no unambiguous evidence for life. The red planet, it seemed, was a dead planet. But the mystery was far from over. For more than 30 years, the contradictory results from the Viking missions remained one of the biggest puzzles in planetary science. On one hand, the labeled release experiment gave a result that looked like life. On the other hand, the other two biology experiments suggested strange chemistry, and the GCMS found no organic molecules at all. This mystery guided NASA's strategy for decades, shifting the focus from searching for current life to following the water and looking for signs of past habitability. The Viking data was a riddle that no one could quite solve until another NASA mission landed on Mars. In 2008, the Phoenix lander touched down near the Martian North Pole. It was designed to dig into the soil and analyze its chemistry, particularly looking for water ice. Phoenix made a groundbreaking discovery. It found that the Martian soil contained significant amounts of a chemical called perchlorate. Perchlorates are a type of salt that is highly oxidizing. This was a major aha moment for scientists who were still puzzling over the Viking results. Suddenly, all the weird and conflicting data from 1976 started to make sense. The Perchlorate Discovery was the key that unlocked the 30-year-old mystery. Scientists immediately realized the connection to the Viking GCMS experiment. To search for organic molecules, the GCMS had to heat the soil samples to a high temperature. It turns out that when you heat perchlorates in the presence of organic molecules, a powerful chemical reaction occurs. The perchlorates act as an oxidizer and completely destroy the organic compounds, breaking them down into simple gases like carbon dioxide and chlorine compounds. This meant that the Viking GCMS might not have found any organics because its own experiment had inadvertently burned them up. Organic molecules could have been in the soil all along. This new understanding also helped explain the results of the other biology experiments. The presence of highly reactive perchlorates in the soil could explain the rapid release of oxygen in the gas exchange experiment when water was added. It could also potentially explain the results of the labeled release experiment as a purely chemical reaction, although that debate continues. The discovery of perchlorates completely changed our interpretation of the Viking data. It reopened the door to the possibility that organic material exists on Mars today, and it made the results of the labeled release experiment seem even more intriguing than before. The search for life was back on the table. 50 years after their launch, the Viking missions remain a landmark achievement in the history of space exploration. They were a stunning technological success, proving that we could safely land and operate complex scientific instruments on another planet for years on end. The engineering and techniques developed for Viking laid the foundation for every Mars lander and rover that followed, from Pathfinder and the Mars exploration rovers to Phoenix, Curiosity, and Perseverance. The data they collected gave us our first true understanding of Mars as a complex and dynamic world, with weather, seasons, and a fascinating geological history. The legacy of Viking's search for life is particularly powerful. The ambiguous and puzzling results sparked a scientific debate that continues to this day. It taught us that searching for extraterrestrial life is incredibly difficult, and that the results may not be a simple yes or no. The mission's conclusion led NASA to adopt a more cautious, step-by-step approach to astrobiology. Instead of looking for life directly, subsequent missions focused on finding evidence of past water and identifying environments that could have been habitable. This strategy has been incredibly successful, revealing that ancient Mars was a much warmer and wetter place, very different from the cold, dry desert we see today. The Viking story continues to evolve. New scientific models, proposed as recently as 2023, have re-examined the Viking data in light of everything we've learned since. Some scientists, like Dr. Stephen Benner, argue that a biological explanation for the labeled release results is still plausible, and may even fit the data better than the chemical explanations. These new ideas suggest that hypothetical Martian microbes might have a unique metabolism that could explain the strange gas releases seen in the experiments. This ongoing debate shows just how rich and important the Viking data is, inspiring new generations of scientists to look at the old puzzle with fresh eyes. Ultimately, the Viking missions are a testament to human curiosity and our relentless drive to explore the unknown. They took us from seeing Mars as a distant point of light to walking on its surface through the eyes of our robotic explorers. They asked the biggest question we could ask of another world, and gave us an answer so complex and mysterious that it continues to fuel our exploration of the Red Planet half a century later. As we plan future missions to Mars, including those that will one day carry humans, we follow in the footsteps of these two pioneering spacecraft. Viking's journey to Mars was just the beginning of our journey to understand our place in the cosmos.